Hello students, welcome to EPG Pathshala. My name is Dr. Priyanka and I teach anthropology at the Department of Anthropology, Dr. Hari Singh Gaur University, Sagar. Today we are going to discuss about models to study gene flow in this module. This module comes under the paper Human Population Genetics. So, before we study this module, let us see what we are going to cover in this module. In this module, we are going to learn gene flow as a microevolutionary force. We will see what are the various models of gene flow. Let's begin. Gene flow is defined as the introduction of genetic material from one population of a species into the another. This process happens by interbreeding. Gene flow introduces new alleles to the population, which further leads to increased variability and new combinations of the traits. In humans, the gene flow takes place by migration. Migration is a process which is defined as a movement of individual from one occupied area to another. Gene flow is the outcome of a migrant contributing to the next generation in their new location. Sometimes migration occurs, but the migrant do not contribute to the population of a new location. Thus, to observe gene flow directly, we not only have to monitor the movement of migrants, but also their reproductive success. That is, their contribution to the population of the new location. If individuals move temporarily and have offspring in the new population but do not remain in the population, we still call it as a gene flow, as the genetic contribution of one population is made into the another. Migration between population has been a consistent feature of hominid evolution since the first dispersal of mankind out of Africa. Regular gene flow between population help us to explain why in the last millennium years speciation has been very rare phenomena. Unlike selection, migration cannot change species-wide allele frequencies, but it is capable of changing allele frequencies in the population. So, it plays an important role in human genetic diversity. Gene flow counteracts genetic differentiation modeled within the framework of larger subdivided metapopulation. An interesting example of how gene flow influences microevolutionary changes in modern human population is seen through African American example. African Americans in United States are largely of West African descent, but there also has been considerable genetic admixture with European Americans. By measuring allele frequency for a specific genetic loci, we can estimate the amount of migrations of European alleles into the African American gene pool. There are several models of gene flow, which includes island models, two-way gene flow model, kin-structured model, and stepping stone model. We are going to discuss about them one by one. Let's understand what is island model. Island model is the simplest place to start with understanding how gene flow works is to examine the case of one-way migration as shown in the island model, or more specifically, the continent island model. This model assumes one very large population where allele frequency changes only slowly over the time, connected by gene flow with a small population where migrants make up a finite population of the individual present each generation. Or in other words, there is a big population called as metapopulation, which is divided into a number of a smaller population. Gene flow from the island to the continent occurs, but the continent population is assumed to be large enough that migration has a negligible effect on the allele frequency. In this case, an island that receives a specific extent of migration from the mainland, but this migration is strictly one directional. That means there is no migration occurring in the reverse way from the island to the mainland. In this way, we can conclude the effect of gene flow from the mainland to the allele frequency of the islands. There are few assumptions for the successful working of island model. These assumptions of island model includes, first of all, no geographic substructure apart from the division into islands should occur. Each population persists indefinitely. No mutation, no selection is operating upon the populations. Each population should have reached equilibrium between mutation and drift. The migrants are the random sample from the source island, and the symbol small m is used to illustrate the migration rate. Migration rate is the proportion of alleles in the island that comes from the mainland from each generation. The figure given in this slide shows the working of island model. Gene flow is one way from the large mainland population into smaller island, such that the allele frequency in the mainland affect the allele frequency in the island but not the other way around. That means there is a migration from mainland to the island, but there is no back migration from island to the mainland. M represent the migration rate in each generation, and it is the proportion of alleles in the island's next generation that comes from the mainland. Because M represent the genetic contribution from the mainland, this means that the proportion 1 minus M of the island's allele come from the island, because the two proportion, one from the mainland and the another from the island, should have sum equal to 1. By now, we know that the gene flow is one way from a large mainland population into smaller island, such that the allele frequency in the mainland affects the allele frequency in the island but not the other way round. M represents the migration rate in each generation and is proportion of the allele in the island's next generation that comes from the mainland. The quantity 1 minus M represent the proportion of alleles in the island's next generation that comes from the island. For example, a value of M equal to 0.1 indicates that the 10% of allele in the island comes from the mainland, whereas the value 1 minus M equivalent to 0.9 indicates that 90% of the alleles on the island comes from the island in the previous generation. In order to simulate the one-way gene flow under the island model, we need to know the initial allele frequency of the island and the mainland. We use the term as P0 to represent the initial allele frequency in generation 0 of the island. We use the symbol capital P to represent allele frequency of the mainland. Because our model only involves gene flow from mainland to the island, there is no gene flow from the island to the mainland. This means that mainland allele frequency P stays same from one generation to the next. In order to calculate allele frequency on the island in the next generation, we have to multiply the proportion of alleles from island and mainland by the respective allele frequencies. Now move towards the another model of gene flow known as two-way gene flow model. The island model focuses on gene flow in one direction, and the allele frequencies does not change in the source population, which was mainland in this case. Although this model fits in some cases a model that allows gene flow in two directions is more appropriate. The two-way gene flow model seeks to remove one flaw of the island model, that is, lack of geographical substructuring. The model assumes equal rate of migration between subpopulation. Here, it is noteworthy that both the models show that even very low rate of migration between the subpopulation are capable of retarding their genetic differentiation. The two-way model mainly shows the equilibrium allele frequency in the two subpopulations are the average allele frequency of the total population when the two migration rates are equal. Such dimension is also important when there are large number of subpopulation, a result that will be useful to remember when considering the process of gene flow in an island model, in combination with another processes of evolution such as genetic drift. The figure shows a simple model of two-way gene flow where the rate of migration is the same in both directions. Here, two populations X and Y exchange migrants and genes at a rate of m per generation. This number represents the proportion of alleles in one population from the other population. Each population receives proportion 1 minus m of the allele from itself. We start with the rate of gene flow per generation m and allele frequencies for population X and Z in the generation T, which we can label as Px and Pz. We can estimate the allele frequency in the population X in the next generation, which is T plus 1, by multiplying the allele frequency of each population by the proportion of alleles provided to population X. This process of extend gene flow can be made to additional generations as shown in the figure, which shows how the allele frequency become increasingly similar to each other. In fact, over several number of generations, the allele frequencies become exactly the same. Eventually, allele frequency in the two populations will be equal without any further change. However, this will happen only when other factors beside migration do not influence allele frequencies. So the change in the allele frequency from migration depends on two factors: the population of the migrants in the final population and the difference in allele frequency between the migrants and the resident. If no difference exists in the allele frequency of migrants and the resident, then we can see that the change in allele frequency is zero. Population must differ in their allele frequencies for migration to affect the makeup of the gene pool. With continued migration, the source of migrating population and the destination population become increasingly similar by their allele frequencies. As a result, the change in the allele frequency due to migration decreases. Eventually, allele frequency in the two populations will be equal without any further change. However, this will happen only when other factors besides migration do not influence the allele frequencies. These factors are natural selection, genetic drift, non-random mating, etc. The next model is a kin-structured migration model. The example of two-way flow show clearly that gene flow acts to make population more similar to each other over the period of time. The higher the rate of gene flow, the more rapidly this convergence will occur. Gene flow is most often considered a homogenizing force that reduces the genetic differences between populations. However, this is technically correct only if we make the assumption that the individuals that migrate and reproduce are a random sample from the source population. This may not always be the case. In some situations, the migration may be related. One way that migraines can be related occur in some small-scale human society when part of a population splits off and then fuses with another population. There are other examples of entire families moving into new populations. When the migraines are related, we call such kind of a migration model as a kin-structured migration model. The genetic effect of kin-structured migration on the genetic differences between population is actually the opposite of gene flow. Gene flow usually acts to reduce the differences between population, while kin-structured migration can slow this process down or even reverse in some cases, leading to increased genetic differences between population. Look at the kin-structured migration among the Sweden agricultural group. Over the period of a time, population split and the resulting group of migrants were typically kin. Using simulations incorporating genetic drift and gene flow based on the demography of the population, Fix found that although kin-structured migration flow would still reduce genetic difference between the population over time, the actual reduction would be less than if the migrants were simply the random sample. Although the island model assumes that the amount of gene flow among all population is identical, a population organized into discrete subpopulation can also experience isolation by distance. The stepping stone model is one such model which approximates the phenomena of isolation by distance among discrete subpopulations by allowing most or all gene flow to be only between neighboring subpopulation. That means gene flow is maximum between the two neighboring subpopulation. This gene flow pattern produces an allele frequency clumping effect among the subpopulation qualitatively very similar. That means two subpopulation were quite closely placed with each other. They share a very kind of similar allele frequencies. Kimura and Viz in their analysis showed that the correlation between the two alleles sampled at the random from two subpopulation depends on two factors. One, the distance between the subpopulations, and second, the ratio of gene flow between neighboring colonies and long distance gene flow where alleles are exchanged among subpopulation at random distances. As expected for isolation by distance, the correlation between allelic states decreases with increasing distance between subpopulations. Interestingly, the correlation between allelic states drops off more rapidly with distance when subpopulations occupy two dimensions than when they occupy single dimension. Another model of gene flow is isolation by distance. The phenomena of decreasing chance of mating with increasing distance separating individual is termed as isolation by distance. One effect of isolation by distance is local change in allele frequency in a population with local regions approaching fixation or loss, akin to the impact of reduction the effective population size. Alternatively, isolation by distance can be thought of a form of inbreeding since restricted mating distance causes homogeneity within the subpopulations. The pattern of genotype in the simulated population bear this out, with an obvious decline in the overall frequency of heterozygotes over time with isolation by distance. But no such decline was observed in case of panmixia. This section is all about the ramification of the gene flow. The effect of gene flow have important ramification not only for the evolution of the species but also for the conservation of the species. Many species that have wide geographical range show variation in genetic structure over the species range. Part of the natural genetic structure of a species could include population subdivision, in which populations are loosely connected to each other by gene flow. Since gene flow is important in maintaining genetic diversity, this feature of population genetic structure must be taken into account by those interested in conserving genetic identity of the species. Finally, migration of individuals into a population may also change the makeup of the population gene pool. If the allele frequency of the migrants differ from the migrant resident population, this phenomena is very much obvious. Migration, or otherwise calling it as a gene flow, tends to reduce genetic divergence among population and increases the effective size of the population. The amount of migration among populations of the same species determines how much genetic substructuring exists and whether different populations of the same species become very different from each other genetically. So students, let us summarize this module. In this module, we have learned that gene flow is defined as the introduction of genetic material from one population of a species to another population by changing the gene pool composition of the receiving population. Gene flow introduces new allele to the population, which further leads to increased variability and new recombination of traits into the new population. In humans, gene flow takes place by migration. Gene flow counteracts genetic differentiation and is modeled within the framework of a larger subdivided metapopulation. The higher the rate of a gene flow, the more rapidly this convergence occurs. Gene flow is most often considered a homogenizing force that reduces the genetic differences between the populations. Gene flow has two major effects on our population. First, it introduces new alleles to the population. Since mutation generally is a rare event, a specific mutant allele may arise in one population and not in another. Gene flow spread these new alleles to other populations. And like mutation is a source of genetic variation for the recipient population. Secondly, when the allele frequency of migrant and the recipient population differs, gene flow changes the allele frequency within the recipient population. The simplest place to start with understanding how gene flow works is to examine the case of one-way migration or the island model, or more specifically the continent island model. In this model, we have seen that one very large population where the allele frequency changes only slowly over time is connected by gene flow with the other small population where migrants make up a finite proportion of the individual present in each generation. To simplify this, it is basically one metapopulation subdivided into number of subpopulation, but the gene flow is happening only from metapopulation to the subpopulation and not the other way around. The two-way gene flow model addresses the issue of geographical substructuring. The model assumes equal rates of migration between the subpopulations. The two-way model mainly shows that the equilibrium allele frequency in the two subpopulations are the average allele frequency of the total population when the two migration rates are equal. In some situations, the migrants may be related. One way that migrants can be related occur in some small-scale human society when a part of a population splits off and fuses with another population. There are examples of entire families moving into a new population or new location. When the migrants are related, we call this model of gene flow as a kin-structured migration. In the next one, we have learned about the stepping stone model. The stepping stone model approximates the phenomena of isolation by distance among discrete subpopulations. It allows most or all gene flow to be only between the neighboring subpopulations. Thank you.